Hey guys, happy Wednesday. Welcome back to another AP Bio stream with Fiveable. My name is Jessica Nadza. I am a 5th year AP biology teacher, so I like to kind of sort of think I know what I'm talking about. And today we are going to talk about some potential FRQ questions you may see on the AP Bio exam concerning body systems. So body systems used to be a full component on the AP exam, not on the AP exam, in the AP Bio curriculum, where it had like its own section and stuff you had to learn. They have since taken that out. However, I have seen a lot of practice questions floating around. Ooh, my hair. And those practice questions reference body systems. What I will say about them is they're not the kind of questions where you have to already know everything about body systems, but they're kind of like ACT questions in that they give you like 10 zillion pages of information that you have to sift through and then utilize what other AP knowledge you should have in order to answer it. And that's cool and all, but like, why not have a heads up and already know the stuff so you don't have to read through everything 12,000 times? That's just me, at least. So, super happy to see some familiar faces or names. Slay, Sierra, Nicole, yay, Selma, I'm so glad y'all are here. Callie, welcome. Let me go ahead and pull up my slides because that's what y'all really came for. Let's get real. My face is not the top priority here. Give me just 1 second. I have lots of screens to share with you guys. So yeah, you're going to see a lot of things today because I have lots of resources because I'm here to help. Yay. And dun, dun, dun. If you watched my last stream when we were here, I was in the middle of a thunderstorm. You'll be happy to know that although there's not a thunderstorm, it is raining outside. And my dog is currently shaking outside my office door. Not like shaking in fear, just like shaking so her collar jingles. So if you hear me go, shh, go away. I'm not talking to y'all. I'm talking to my dog. I promise. Because I want you guys to be here because I want to tell y'all about body systems. So let's go ahead and get started with it. Review. And the review, there's only like 2 or 3 slides for each of those review sections. I am not here to give you the breakdown on everything cell transport, everything cell signaling, everything nervous system, everything immune system. Not what I'm here to do. What I am here to do is talk about, you'll see it right on the bottom there, those practice FRQs. And in order to answer those practice FRQs, it's important to me to make sure that you remember the background knowledge necessary to answer that. So that's why we're kind of going back into cell transport and cell signaling, not to like completely restart them, but just to make sure that we don't have any misunderstandings when we go to answer those FRQs. This is the knowledge you will need to answer those FRQs. I would like to point out though, in case you hadn't already noticed, when we're talking about body systems, we have to go all the way back to like unit 2 with cell transport. Most FRQ questions are going to do that. It's not going to be one FRQ that is all about photosynthesis and all the questions are about photosynthesis. No, they're going to tap into every single unit that they possibly can because they want you to answer lots of things. If you've been in the game for the past week or 2, you know that the AP exam has changed a little bit just for this year. So want to make sure we're all up to speed on how that's going to work. All right. So let's take a look at cell transport review. La, 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 la. Can anyone tell me what this scary alien landscape is? I've used this GIF several times, but I always like to check and see. What's that scary alien landscape looking thing? What is that? Cell membrane. Very good, Sierra. So this is the cell membrane. This is what your cell membrane looks like. A scary alien landscape. Yes, all of ours looks like that. What's that yellow thing all along the middle? Phospholipid bilayer. Very good. Okay. So that yellow thing here all along the middle, let me get my little laser pointer out. That's very good. A phospholipid bilayer. And this is what separates the outside of the cell from the inside of your cell. We know this is the inside because it's got like a mitochondria thingy down here. And then what are these little thingies that are sticking in and out of the membrane? What are those? Proteins. Very good. Okay. So we say that your cell membrane is like a fluid mosaic because it is studded with different proteins and materials. That's the mosaic part. And we say that it's fluid because it's all wibbly wobbly alien landscapey. Okay. So that's the review on that actual structure. The rest of that, don't really need you to know, but I do need you to know. I do need you to know, but not for these specific problems. I mainly need you to know that there's all these proteins sticking out. What do we use those proteins for? Ooh, someone already said it. Very good, Soleil. Transport proteins. Get stuff in and out. Because can anything just walk through the cell willy-nilly whenever it feels like it? Some things, yeah. What kind of things? What kind of stuff is going to be able to just go through there? Some small non-polar things. Very good, Soleil. Okay, so typically things like oxygen, small gases, stuff like that. Nicole, very good to you as well. Woohoo! So now that we know that, let's talk about how that stuff is going to move through the cell membrane. So right now what we're seeing here, we've got we've got our phospholipid bilayer with our little phospholipids with their heads and their tails. And they're amphipathic. That means one end is polar, one end is non-polar. And that's why they line up the way they do. So here we can see there's a bunch of different stuff in here. We've got some glucose molecules, we've got some potassium ions, and we've got some sodium ions. Why can't glucose just pass through the cell membrane? Like, it's sugar. We need it to survive. It's too big. Very good, Soleil. Okay, well, what about like potassium and sodium? Why can't it just pass through the membrane if it's so small? Ions. What does ions mean? Does that mean it has like eyes on its head? What does ions mean? They're charged. Very good. And as I said a moment ago, phospholipids are amphipathic, which means part of them is charged, part of them is not charged. They don't like charged stuff passing through, so those ions can't pass through. So instead, they have to roll on through some transport proteins, which is woohoo. So we've got sodium, potassium coming through here. What do we call this particular protein where the sodium and potassium is passing through on the left? That's an important one. Sodium potassium ion channel, sodium potassium pump, proton pump. Yes, very good. All of those are accurate. And you're going to need to know the sodium potassium pump for when we talk about neurons, especially this thing called an action potential. So keep that in mind. You can only let one sodium, or hang on, let me double check. I have to make sure I'm reading it properly because if I tell y'all wrong, I will stay up all night feeling guilty about it. So we can see that potassium is entering the cell and sodium is going out of the cell. Okay? So one's going in, one's going out. But they both have to be doing their thing, as in one is going in, one is going out. Otherwise, it doesn't work. You can't just have one going in and that's it. What else do we see that's essential to that sodium potassium pump? It uses ATP. Very good. It uses energy. So that means it's going against the concentration gradient, which means, is it active or passive transport? Active. Very good. Okay. So it's active transport. It's using ATP. It's going against the concentration gradient. Now, when I taught sodium potassium pump at the beginning of the year, I always told my students it had to do with a balance of electrolytes. So, for example, like I've got some Gatorade here. I've had a little bit of a headache all day, which happens to me whenever the weather changes, and our weather here has been just nuts. And I don't know, for some reason, drinking Gatorade makes me think it's like the cure-all for headaches. But whenever your electrolytes are in balance, that can cause headaches. That's why when you're dehydrated, you can get a headache, things like that. And it also can relate to muscle cramps if you don't have the proper ratio of electrolytes in your body. Electrolytes coming from the Gatorade. I think of a headache as just a giant muscle cramp in my head. So that's why I drink a bunch of Gatorade when I have a headache, I guess. The reason I'm telling you all these minuscule details about my life is previously when we've talked about sodium potassium pump, we were talking about like muscle cramps and electrolyte balances. Now, how that's going to relate to neurons actually will relate to muscle cramps and things like that. So we'll be getting into all that, but I want you to remember that sodium potassium pump. Does anyone have any questions about this review so far? Drink my Gatorade. I have to put it on ice. That's why it's like, looks like alien juice over here. I don't see any questions. I'm going to give you all 3. So is there more sodium in or outside the cell? So keep in mind it's active transport, which means it's moving against the concentration gradient. So we can see sodium is the green dot. Let me get my little laser pointer. And sodium is moving out. If it's moving against the concentration gradient, that means there should be more sodium in, or more sodium is out, and it's moving out, and then it's got that imbalance. That's why it's active transport. Same thing on the reverse for that potassium. Does that help, Soleil? I feel like I kind of butchered that a little bit. Okay. If I ever need to re-explain anything, guys, please don't hesitate to ask. My students tell me all the time that I talk 10,000 miles per hour, and sometimes I get my words mixed up. So no shame in telling me, that makes no sense. Please explain it again. Okay, but I'm going to go ahead and go on. Let's talk about cell signaling here. So before we start cell signaling, I'd like to share a TikTok with you because I have nothing to do for several more hours a day than I am used to. And therefore, I spend way too much of my time on TikTok. And I found this TikTok, and it reminded me of cell signaling. So you may have seen it. It's called like the emoji challenge. Let me see if I can get it to play. Okay, so that was a really short emoji. I just thought it was cute. But the point of the emoji challenge is, so like it generates a bunch of emojis and like hand signals, and you have to copy what it's doing to the beat of the music. So it's a signaling game. And your body is the exact same way, because whenever it's exposed to something, some ligand, and it enters the receptor, the cell in your body has to do a certain thing. So like, for example, if I drink this, it's got a little bit of sugar in it. Not a little bit. It's got a decent amount of sugar in it. So when I drink Gatorade, it's got some sugar in it. Whenever that sugar reaches my liver, it tells my liver, hey, we got to start absorbing this stuff so that the blood sugar doesn't go through the roof. My body reacts to that. If I am exposed to pollen because I have ridiculous allergies, it enters my nose. My nose says, oh boy, oh boy, oh boy, time to start sneezing to get the pollen out. Like, when your body is exposed to something, it reacts to it. Just like in the emoji challenge, when you see that emoji, you have to copy it in some way, shape, or form. So we can do cell signaling through a couple of different ways, though. There are 4 specifically that we talk about on the AP exam. There's endocrine, and that's probably one of the most widely used that they give examples of, at least. So endocrine is like hormones. That's when it's got to travel all the way through the body. So for example, if, let's say I'm having a hard time thinking, oh, someone already brought one up, insulin. So insulin, whenever that is released, I believe insulin is released from which organ? Tell me. Where do we release the insulin from? Tell me. I know y'all know. It starts with a P. Pancreas, very good. So whenever I drink some sugar juice, my pancreas is triggered by the sugar, the glucose, and then my pancreas releases insulin, which travels through my bloodstream until it hits my liver. And my liver is like, that's a lot of sugar. And so my liver will release different chemicals that then tell my body, okay, time to take up all the sugar. That's endocrine. It's hormonal. It's traveling all throughout the body. Now, there are other types that we use as well. The first one is autocrine. This is when a cell will actually signal itself. You see this often in cancer cells when they will tell themselves, okay, time to grow, even though they're not actually supposed to grow. They're just growing uncontrollably, which is the definition of cancer. Paracrine is when a couple of cells are like next to each other, but they're not necessarily touching. So that'd be like if a cell in my hand told the cell next to it in my hand, hey, time to grow, divide. I don't know. And then the last one, contact-dependent signaling. These are when cells are actually touching one another. What's another word for that contact-dependent signaling? It starts with a J. Who can tell me what it is? Juxtacrine. Very good, Sierra. So juxtacrine is when the cells have to actually be touching each other. This juxtacrine is going to be extremely important for immune response because you've got elements of your immune system, white blood cells, that literally just creepy crawl down your bloodstream looking for stuff that's not supposed to be there. And so it's kind of like, you remember when you were a kid and you would clean your room, but your mom had to tell you several times, hey, clean your room. And so whenever you're like, look, Mom, I cleaned it, she'd walk in and she'd kind of look around like looking for something that you didn't do. Like my thing was always when I was a kid, I was really bad. I hid everything under the bed. So my mom would walk in and kind of look around and then she'd just look under the bed and be like, you didn't do it right. Do it again. White blood cells are kind of like your mom when you cleaned your room. They're walking around, looking, checking things out, looking for anything that's not accurate and not where it's supposed to be. And they're very, very good at it, which is a good thing because that keeps our immune systems alive so that we can fight off most infections. So they're going to use that juxtacrine system. They have to touch something in order to react with it. So whenever this is all happening, though, whenever they're touching or passing on things, whatever it is, I want to talk about specifically ligands and how they go into a receptor and they have a response. We call that a cascade. You don't need to know all of that for the purpose of these FRQs. If you don't remember that, you may want to go back and watch the signal transduction videos. But so a ligand is anything that is causing a response. So for example, it can be the sugar in my Gatorade. It can be the ibuprofen in my Advil when I have a headache. It can be the all sorts of different stuff. I mean, you have tons of hormones. You have tons of different things you react with. It could be the molecules in the pollen once it hits your nose and makes you sneeze. But you have a receptor that recognizes it. And that tells it, the ligand is the signaling molecule. Yes, very good, Soleil. The ligand hits the receptor, kind of like an enzyme and a substrate. But let me be clear. While they have to fit together, they are not the same thing. They're not necessarily catalyzing some reaction to save energy. And the ligand or the substrate, whatever, isn't necessarily going to be reused. So while it's the same mechanism, two things that have to fit together, kind of like Legos, it's not the same process. So let me make that clear. They just kind of look alike. That's not confusing at all. So once that ligand hits that receptor, it's kind of like whenever you take something, like say this calculator, and I put batteries in it. Those batteries turn it on, and then it goes ding, and it can do the thing. Whenever the ligand hits the receptor, that's kind of like putting batteries in. It turns the receptor on, and it goes ding, and it does the thing that it's supposed to do. And it sends some signal all throughout the cell and says, wee-hoo, 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 it's go time, here we go, la-ha. That's the sound all your cells make. They go, wee-hoo. So they'll have some cascade. If you are interested in knowing the specifics of that cascade, I strongly recommend you go back and watch the signal transduction videos. I don't have time to go into that right now. The main thing you need to know is the ligand hits the receptor, that turns the reaction on, and then it will elicit some response from that cell, whether it's telling the cell you need to divide, or telling the cell you need to go hide because we have an infection, or telling the cell you need to explode. We don't want you anymore, which your cells do sometimes. It'll have some response. I'm not going to go into the mechanics of those reactions right now. Like I said, we don't have time. You just need to know when the ligand hits the receptor, it goes bing, and it turns on. Now, that can be very, very, very specific when you have very specific types of cells. So for example, here on the left, I have a specific type of cell. What is this specific type of cell here on the left? Or two cells, I guess I should say. Neurons, very good. Now, neurons in this instance don't look like the typical round cells that we see. You're kind of like, what is that? But that's okay. That's how they pass on signals. So for example, I have, because I'm so excited to be teaching right now, I got some endorphins flowing because I don't get to do a lot of teaching face-to-face with kids right now. So now I am, and I'm like, woohoo, I'm so excited. So there are endorphins, happy chemicals, in my neurons, and they pass on to the next door neuron and tell it, hey, be happy. And that one's like, okay, let's do it. And it goes round and around my brain, and here I am. Okay, let's look a little specifically at that. And you may have done this lab or this virtual lab in class before, but I really, really like this one. And so we do it every year. But you've got these neurons in your brain, in your body. These are your nervous system. So you're really going to need to know this for when we get to the FRQs in a minute. So these cells are, again, they look like aliens. Like you look at them and you're kind of like, what even is this? Neurons have very specific parts that do very specific jobs, okay? And they can live in your brain, but they also live all along your muscles because your neurons have to fire to be like, okay, now clench your fist. And then you're like, brrr. And then they have to fire to be like, wave your arm in the air. And you have to go, woo. And then they have to be like, put your hand on your head. And you're like, okay, you have neurons that tell your body parts, hey, do this, do that, do whatever else, okay? But in order to do that, they have to use this very specific structure. The main two components you're going to see are these, let me get my laser pointer, dendrites and axons. So the dendrite you can see kind of looks like this wide spider webby thing. I like to think of it like a catcher's mitt. So like if you've ever seen baseball or softball or whatever, you know, they wear a mitt. The reason they wear a mitt instead of using their hands is, one, baseballs and softballs are hard and that would hurt. Two, they are a lot wider than your actual hand. And as a result, that gives you more surface area to catch the thing you're trying to catch. Same concept goes here with the dendrite. By having all these little projections that stick out, they're a lot more likely to catch a signal. They've got little receptors on all these ends on the dendrites that are just waiting to catch some ligand that flows through, whether that's an endorphin or another brain chemical, whatever it is, dopamine, serotonin, norepinephrine. You don't need to know those words. If you know them, great, but you don't have to. So they've got this like catcher's mitt wide end. But once it receives that signal, it has to send it down this line and it goes woohoo, 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 woohoo until it hits the axon. Now this axon is going to be connected to another dendrite. So kind of like we saw here, axon dendrite. And it's going to pass that ligand on to the next cell. It wouldn't hurt to know that here you've got these little yellow thingies all along the axon, which is this long skinny part. Those little yellow thingies, just so you know, those are composed of fat. They call them myelin sheaths most commonly. That myelin sheath is fat. So it's kind of like... So I've got a lot of snacks in the way. So for example, here's the cord. I'm going to make this bigger so y'all can see for a second. Okay. So here's my charging cord. If you've ever noticed, cords have these things that wrap around them. So this is one of those ones I got on Amazon. It's got like a cloth covering over it, and that's supposed to prevent it from like breaking off or whatever, as opposed to, say, the typical Apple ones, which we all know sometimes have that tendency to go and they kind of break open. This one hasn't yet, thank goodness. I hope I didn't just make it more likely to break. But we have this wrapping as insulation, because if we didn't have it, would the cord work? Would it transmit the energy or the power for the battery anymore? No. So this is like insulation. If you've ever had like a puppy or a small child in your house who like chews on these things, you'll notice that once they're opened up, you can see all the little wires inside. And usually these cords won't work anymore because the signal from the wire gets lost without that insulation to keep it in. Same concept goes for those myelin sheaths on your neurons. So those are insulation because there is literal electricity in those neurons. And when that insulation goes away, the signal, the electric signal in your neuron gets lost and it can't pass it on. That's actually the cause of several neurodegenerative disorders. The brain itself is fine, but the myelin sheath, once it starts disintegrating, the brain gets very unhealthy because it can't pass those signals where they're supposed to go. So that's why sometimes you may see like jerky movements or just the loss of ability to move. So once you see, okay, so once you know how all that works, now, so we've got a dendrite because this one is releasing something, or sorry, I've got it backwards. We've got an axon because it's releasing something. And then we've got a dendrite right here that is receiving something. So what I'm going to show y'all now, this is one of my favorite labs I do with my students. It's called Mouse Party. You can look it up. But basically, it's an interactive way of seeing like how those chemicals and neurons and stuff in your brain look. So I'm not going to spend a ton of time on it because we've already been here close to 30 minutes and we've got a lot more to do. But I do want to show y'all just a little demo of it because it is cool. So like, okay. So we've got this game called Mouse Party, and it's real interesting. Here, let me make it bigger so y'all can see it. So Mouse Party, you have all these lab rats who we do an experiment on or whatever. But so what they do is all the mice have been exposed to illicit substances. And so we can go in and look at their brain and see how it impacts their brain, because the reason that illicit substances like drugs affect your brain or affect you is because they directly interfere with your nervous system. So for example, here, I can kind of swirl around. I can pick a mouse on LSD, cocaine, I'm going to pick this one. He looks like he's kind of awake. So we go over here. Can I turn this music down? Oh, well. Okay. So the whole reason I like this, though, is it goes...
Serotonin transporters are responsible for removing serotonin molecules from the synaptic cleft after they have done their job.
So the whole reason I like this is you can see the molecules that are involved in each specific substance and how it messes with the brain. So this is just a cool way to see how it works. So here we have the axon, and here we've got a dendrite that is received. So we've got these transport proteins, serotonin transporters, okay? They have the serotonin neurotransmitter. So these are responsible for removing serotonin from the synaptic cleft after they've done your job. Just for context, serotonin is the neurotransmitter that makes you happy. It makes you go like, la, la, la. Basically, when I was in high school, I learned about serotonin and norepinephrine. So there was like happy Sarah and negative Nora. Norepinephrine, negative Nora, is kind of stereotyped as like the unhappy neurotransmitter. It causes you stress, which to a degree is good. If you were never stressed, that may interfere with your ability to like take tests because you wouldn't study. Serotonin is like the opposite end of the spectrum. It keeps you happy, things like that. So when you take, for example, ecstasy, in this example, the
ecstasy mimics serotonin and is taken up by serotonin transporters. In fact, ecstasy is more readily taken up than serotonin itself.
So ecstasy basically mimics serotonin. It's the same ligand shape. And so it gets taken up into the dendrite because the dendrite thinks it's serotonin, thinks it's going to make it happy. But the reality is it overloads your brain, and then your brain's like, I'm so happy. But then when you don't have any more ecstasy, your brain's like, oh, I'm not happy anymore. So the point of this is you can see how the neurotransmitters are moving through the brain. So I would strongly recommend...
This interaction with ecstasy alters the transporter. The transporter becomes temporarily confused and starts to do its job in reverse. The transporter starts transporting serotonin out of the cell.
So this just gives a good example of how stuff moves through the neurons. So I think it's a really good visual aid. It's linked in the slides, so you'll be able to find it whenever I publish the slides. So definitely take a look at it. It's just a really good way to get familiar with the neurons if you're not already familiar with them. Okay, let's go back to our slideshow though. So we kind of already saw there in Mouse Party how the axon will pass on these ligands, which are then picked up by the dendrite. And those can be various different molecules depending on what they need to do and in what part of the brain. So like, you know, you have certain parts of your brain that are associated with memory, happiness, stress. They're going to react with certain neurotransmitters to do that. And so when they do that, they have this thing called the synaptic cleft. And that's what's going to be pumping all that stuff out and then passing it on to the next. Yeah, I can't talk right now. Passing it on to the next neuron. I want you to notice, though, this thing called a voltage-gated Ca++ channel. What is that Ca++? What is Ca++? Calcium. Very good. So that calcium, it's in a voltage-gated channel. What does voltage mean? Energy, electricity. Very good. Remember what I said earlier. Neurons are literally electric. Just like there's electricity that flows through this cord when I plug it in, there are teeny tiny electric signals flowing through your brain constantly. That's why when they plug you up to EKGs and stuff, they are measuring the voltage in your brain, okay? So we have this voltage-gated calcium channel. That transport protein, okay, it is activated by electricity that makes it release whatever the stuff is that needs to go on down to the next dendrite. Any questions so far about neuron structure and how they work? Okay, I'm going to go on. Again, that was the abbreviated version, but that's just what you need to know to answer the FRQs we're going to talk about. Okay, now's the part where we really get into how that electricity works. So you can see here, crack the knuckles because this is the big part. Woo. All right, so you can see here there's a bunch of different stuff going in and out and in and out. Sodium and potassium, okay? And whenever you change those concentrations of sodium and potassium, that changes the voltage or the amount of electricity in the neuron. And once that change hits a certain level, we reach this thing called an action potential, and that's what will cause the voltage-gated proteins to open and shut to let neurotransmitters in and out. Okay? There's a lot to that, but that's the basic of what you need to know. And if you really want to get the basics of what we need to know, then I will show you yet another simulation. This is another free one on the internet. It's called FET. I'll have it linked in here so you can see it. And so in here, what you've got, this is like a cross section of an axon. So it's like we chopped it in half so you can see it. And so let me make this a little bigger for y'all. What is action potential? No worries, Soleil. That's a good question. I should have answered that already. So action potential is that electricity moving through the neuron, okay? And it changes depending on how much electricity is moving, and how much electricity is moving depends on how much sodium, potassium, and stuff is moving through the axon. Does that make more sense? Okay, I'm going to zoom this in for you guys. So this is the cross section of it. Oops, I can't do that on here. I have to do it on here. Okay, so I'm going to zoom this thing in. So you can see we've got sodium and we've got potassium, and they are flowing in and out of their respective channels. Okay, they have what are called leak channels. These are just channels that are open all the time. So stuff can pass through no matter what, but it has to go along the concentration gradient. Then they have gated channels. These are going to be the ones associated with active transport when they're going back and forth. So I want you to notice here we've got these charges, okay? And I'll show you all that in a minute. Whenever those charges change, that's the action potential. That's the electricity that's going through the neuron and making it fire. So I'm going to stimulate a neuron. So think of it this way. It'd be kind of like if you, let's say you raise your hand, hold your hand, now take your other hand and you go pow, and you pop your hand. You feel that, right? Your hand reacts, it goes, no. Let's say I did that to your hand, or you do that to your own hand. I can't do that to your hand. We're stimulating those neurons. Pew! So you can see that purple line where the electricity came through, and all of a sudden that sodium and potassium starts flying through those gated channels. Okay? I'm going to zoom out a little bit so you can see it kind of come all the way through. Watch again. It goes zoom, and then everything flows in and out super, super fast. Okay, that's the electricity flowing down your axon. Remember, it's insulated by that myelin sheath. That myelin sheath is keeping the electricity insulated so that that signal can pass down your neuron, then go to the next neuron, to the next neuron, till it reaches your hand, where your hand is like, no. Now, if I zoom back in, So let's take a look at these channels. I'm going to make this one go in slow motion. So as that charge comes down, whenever the charge hits, pew, you can see the channels open so that stuff can get in and out. And you can see the charges flip-flop. But after a moment, they close back again and the charges flip-flop back. That's the action potential. If you really want to get scientific with it, we're going to reset this. Oops. Let me bring it back in. We have this thing called a potential chart. This is the same chart I showed you earlier. We're going to stimulate that neuron. We're going to go pew, and the neuron goes, ah. And you can see this line where it was stimulated. That's where the neuron reacted. And then over time, it slowly goes back to normal where it was. That's all an action potential is. That's all you need to know about it in the terms of this. But I like to give people the visualization so that when they see it like on the test, they're not just like, it's the potential for action. Instead, you can kind of have some image of it in your brain and be like, oh yeah, that's when it goes pew. That's the sound I want to play in your head whenever you read action potential. Does anyone have any questions about that before I move on? It's okay if you do. It's a lot to take in. Just quickly recap. Yeah, Callie. Okay. So I'm going to back out of this so we can kind of get a full view of it. And I'm going to go in slow motion. Okay. So you raise your hand, tap your hand recognizes that. It's like when you're at the doctor and they take the little hammer and tap your knee. You stimulate that neuron and it runs down the axon, goes boom. When it is stimulated, that's when that sodium and potassium goes in active transport through the neuron. That's the electrical charge that it feels because those charges change. You can see that right here. Okay. And then it sends the signal down the rest of the neuron so that then it can go to another neuron and another neuron and another neuron. Whenever these gated channels open and close, I'm going to X out of this, go back to the slide. So if you remember, I've got the big screen, make me the little screen. So if you remember here, remember voltage gated channels right here down at the end of the axon. So whenever they get that pew that hits, that causes them to open so then they can pass on the next neurotransmitter to the next dendrite. And it can keep, because what happens is those neurons just keep going like a little railroad through your body. Does that make more sense? You good? Okay, awesome. All right, gotta hurry. We only got like 20 minutes left. Sorry. Okay, let's talk real quick about the immune system. This one's gonna be really quick though. So what we've got, a white blood cell, some red blood cells, and a little bacteria. Your white blood cell, like I said earlier, is like your mom looking through your room when you said you cleaned your room, but she's like, I don't believe you. You didn't clean your room. And it just kind of swims around looking for stuff. So like you can see this white blood cell is looking around for an antigen until he catches it. White blood cells are like Liam Neeson. They're like, I will find you. Taken? Did anyone see that? It came out when I was in like high school. So it's like 10 or 15 years old. So it's okay if you haven't. But they are kind of the kickstarters behind the immune system. If you don't have the white blood cells, it's really hard to activate the rest of the stuff. So let's look at what that looks like here. So these are two separate charts, but I put two of them because I think it helps to have multiple visualizations of things. So I'm going to wait for that little gray screen to go away. Okay. So what you've got, it starts with an antigen presenting cell, an APC. Okay. So what happens is when your little white blood cell is trekking around through your blood cell and he finds some antigen, some bacteria, something that's not supposed to be there, the white blood cell will literally just glomp it up, eat it, and then make it into teeny tiny pieces. But then, because white blood cells are a little bit nasty, they will wear the remains of that antigen on their sleeve. And they become antigen-presenting cells. So they've got the remains of whatever bacteria, a little piece of it. You can see that here. Okay. But why that's important is they will then go to their friend, the T cell, and present that antigen, the antigen-presenting cell, to the T cell. And the T cell's like, oh, thank you, I'll go find them. And then the T cell will go off and like keep going with the process. So you can see here in this second diagram, those T cells are going to go and activate T helper cells, who can then activate more macrophages, those are white blood cells, or B cells, and those B cells will then get antibodies. Antibodies are kind of like the foot soldiers of your immune system. They just float around and they're like, I see a problem, get it. It's what they do. It's kind of like whenever you've presented for a cold, you will eventually produce antibodies that will go and attack that cold. Now, these helper cells can also go and get killer T cells, also called cytotoxic cells, and these will continue to go and just directly by themselves tackle that antigen. So you don't need to have all of this memorized. Let me make that clear. That's a lot of information. And like I said earlier, College Board doesn't have like a full section on the immune system anymore. They used to, but they got rid of it. However, I've seen so many practice questions and old questions that talk about stuff like this. They give you all of this information. Like they would summarize, this is what a T cell does. This is what a macrophage does. They would tell you in the test, but that's like 4 minutes of you having to read through a paragraph instead of you just already knowing it so you can just go and answer the questions. Does that make sense? So that's why I choose to go ahead and give y'all this instead of not giving y'all this. Letting you be prepared. It's a little extra, but it's okay. Any questions about this slide before we get into the FRQs? We've got about 15 minutes left to go over those. Three, two, one. Okay, let's go ahead and go. So when I was making the FRQs, or when I was finding them, so these are all publicly released FRQs on College Board. Like I have access to the test bank that College Board gives us for AP Bio because I'm an AP Bio teacher, but I'm very, very careful to make sure everything is like publicly released. Like you can Google these and find these and like, that's fine. But when I was trying to format them, it was really weird putting them on a slide, especially when they had a lot of pictures. So it was easier for me to just put these in these PDFs. You should be able to get these PDFs from the links whenever the slides are released. So don't be like freaking out trying to get everything. You'll be able to get it. So today we're going to look at practice FRQ for neurons and a practice FRQ for immune response. So let's go ahead and start with the neurons. Let me make sure y'all can get that because that's what you need. I hope I put the right one. Not the scoring guide. Come on. It's taking its time because my internet has been super slow lately. I'm just really glad that I was able to do this stream. It says waiting for Google Drive. To which I say, I'm not patient. This is awkward. Let me see if I can get the other one to load real quick. Okay, there it goes. Perfect. So let's make this big. Okay. So this is an example nervous system question that I've seen before. This should be very familiar to what we just talked about. That's why I made you go through it. So you can see we've got a dendrite. It's going to release that neurotransmitter. We've got an axon. It's going to release the neurotransmitter. We've got a dendrite that's going to receive it. We've got a synapse. We've got the cleft, all that stuff. And here it's going to tell you that acetylcholine is a neurotransmitter that can activate an action potential in a postsynaptic neuron. A lot of big words. Main thing you need to know is this is acetylcholine. It's a neurotransmitter. Okay. Then we've got the action potential. So when you get to the top of this line, like we talked about, that's when it's firing. That's when it goes boom, and all the calcium, or not the calcium, the sodium and potassium go in and out super, super quick. So we've seen that. That's not new. Okay? And this happens super quickly in these milliseconds. So far, so good. I'm going to go super slowly on this, guys. Okay. Thank you for the thumbs up. It's very affirming. So here it says, describe the immediate effect of the neurotoxin on the number of action potentials in a postsynaptic neuron. Predict whether the maximum membrane potential of the postsynaptic neuron will increase, decrease, or stay the same. It's a lot of words, a lot of syllables. Part B, the researcher proposes two models, A and B, for using acetylcholine, an enzyme that degrades acetylcholinesterase, an enzyme that degrades acetylcholine, to prevent the effect of the neurotoxin. In model A, ACHE is added to the synapse. In model B, ACHE is added to the cytoplasm of the postsynaptic cell. Predict the effectiveness for each proposed model. Provide reasoning to support your predictions. So if you had one of my previous streams, you know we've already talked about how to talk about FRQ questions. So first things first, we want to go and find our action verbs. What are the action verbs for part A? Describe and predict. Very good. What about part B? Provide reasoning. There's one more. Predict. Very good. Okay, so we've got a, let me highlight here. Describe, predict, provide reasoning. Okay, like I was telling people last week, the AP exam this year is not going to give you points for anything you could find on the internet. That is, they're not going to ask you, what is action potential? Because you could just find that in a textbook. And to prevent cheating, they're not asking questions like that, at least not that they'll give you points for. Instead, it's all this predict, provide reasoning, justify, describe. So this is very aligned with what the AP test will look like. Okay? I would give you all time to answer and then come back to it, but for the sake of time, we're going to go ahead and start looking at what these answers are. So if you want to like tune out for a minute, that's totally fine. But we got to keep on rolling. Actually, you know what I'm going to do? I'm going to go ahead. So in case you are one of those people who wants to go over the question and have a chance to answer it before you see the scoring guideline, I'm going to go ahead and look at the second question really quick. That way, if you don't want to hear the answers, you want to be able to like stop and work on it first, you can just stop the stream and come back and rewatch it whenever the rewatch is uploaded. And that way you still get the answers and the explanation, but you get a chance to work through it. But we've only got about 10 minutes, so I just want to make sure everyone gets everything they need. So if you just want to watch it, that's totally cool. This is all about your learning style. So I'm going to go ahead and go to the immune response question. This one's going to be a little longer. Okay. So here's the immune response one. This is the one that really wouldn't fit in the slide. So this is why I had to do it like in a document. And again, this is free information. Like it is publicly available according to College Board, but it will be linked into the slides so you can get it later. Okay. So here we've got a bacterial infection. So we're looking at the immune system. We've got inactive and active stuff. We've got pores. We've got these weird things that we don't know what they are. A lot of stuff. Don't let that make you panic, okay? There's a lot going on, but you can process it. You just have to take your time and go through it. So, some pathogenic bacteria enter cells, replicate, and spread to other cells, causing illness in the host organism. Host cells respond to these infections in a number of ways, one of which involves activating particular enzymatic pathways, as seen in Figure 1. So Figure 1, all this is just an enzymatic pathway. Cell signaling, like we talked about. Cells normally produce a steady supply of inactive caspase-1 protein In response to intracellular pathogens, the inactive caspase 1 is cleaved, that means split, and forms an active caspase 1. Step 1. So you can see right here, step 1, it goes from inactive to active. All it has to do is lose that little black dot. That's it. It's like flipping a light switch. We're saying turn on, turn off, turn on, turn off. Ignore my Scrabble game with my mom. So then active caspase 1 can cleave 2 other proteins. So that active caspase will go on, cleave interleukin to turn it on, cleave gasdermin to turn it on. That's all that's happening here. There's a lot of words, but if you read through them carefully, you can figure it out pretty fast. So researchers created the model in figure 1 using data from fractional studies. When they say fractional studies, that means they like cut stuff up into pieces to see what they would do. In the experiments, various parts of the cell were separated into fractions, fractional studies, by mechanical and chemical methods. So maybe they put it in a blender, maybe they exposed it to some enzymes. Specific proteins known to be located in different parts of the cell were used as markers to determine the location of other proteins. The table shows the presence of known proteins in specific cellular fractions. So if you've got a whole cell sample, this is our control. It's got all of the enzymes. So these all end in ACE. They're all proteins, or these two end in ACE, but all of these are proteins. So in fraction 1, fraction 2, fraction 3, fraction 4, you can see what is present. So if we go to part A, it says, describe the effect of inhibiting step 3 on the formation of pores and on the release of interleukin from the cell. Part B says, make a claim about how cleaving inactive caspase 1 results in the activation of caspase 1. A student claims that pre-infection production of inactive precursors shortens the response time of a cell to a bacterial infection. Provide one reason to support the student's claim. Part C, a student claims that the NFKB protein is located in the cytoplasm until the protein is needed for transcription. Justify the student's claim with evidence. Identify 2 fractions where N-terminal gasdermin would be found in the cells infected with pathogenic bacteria. Part D, describe the most likely effect of gasdermin pore formation on water balance in the cell in a hypotonic environment. And part E, explain how gasdermin pore formation and interleukin release contribute to an organism's defense against a bacterial pathogen. Oh my goodness, those are so many words. We're not panicking over all those words though. We're thinking through one step at a time. So this one is really nice because all of the action verbs are already in bold. Obviously, this is not what your test is going to look like this year because it's got A, B, C, D, E. This test is only going to have A, B, C, D. But the types of questions are pretty well aligned with what it's going to look like. There's no identify, there's no construct a graph. It's all describe this phenomenon, explain this, make a prediction, justify, Okay? So now that we've gone through all that, we've got about 5 minutes left. I want to go over the answers. If you want to work through these independently without me explaining it first, it is totally okay if you want to hop off the slides and just come back and watch the review. But I do want to show y'all one more thing before we go, in case any of you hop out. In case you have not heard, Fiveable is doing these awesome things called Fiveable Olympics. Whoop, whoop. So Fiveable Olympics is for those of us who need some competition, but maybe don't have a lot of outlets for it right now because, you know, coronavirus. So what we're going to see here, let me show you. So the Olympics that we're doing, they start in May. Whoop, whoop. So you can put your email in if you're interested. Again, these will be linked to the slides. So there's going to be all these different games, all these activities you can do, and you get points for studying. Okay, and you do this in teams with 4 people, so find your 3 best friends, woohoo. Or you can compete with your class if your teacher is doing it, and you win prizes. So I don't know about y'all, but I like prizes. So yeah, so just wanted to throw that out there in case people are going to hop off. Okay, but let's go ahead and talk about the answers now, so that then you can earn prizes with Fiveable Olympics. Whoop, whoop. So first things first, let's look at the scoring for the neurons. Share this. I don't know if you can do it individually or not, to be honest. I would sign up for the wait list and then they should give you more information on if you can or not. Okay, let's look at Okay. So here's the scoring guide for the nervous system. So you can see here, got the questions. Describe the immediate effect of the neurotoxin on the number of action potentials. Okay. So this neurotoxin, it said, was that acetylcholinesterase or whatever. In order to get 2 points, you had to say 2 things to get that: describe and predict. You had to say that the neurotoxin will increase the number of action potentials. You had to predict that the maximum membrane potential of the postsynaptic neuron will stay the same, okay? These action potentials don't change. They don't, because there's only so much charge that can go through here. But in terms of this part, the neurotoxin will increase the number of action potentials. You can have more of these, but the shape will always look the same. And the reason is, if you're exposing it to this toxin, it's going to mimic that pattern. So you're going to get more action potentials because it's going to go fire, 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 fire. Part B, that was where it asked us, which one do you think is going to be the Part A model or the Part B model? And then provide reasoning why. So for Part B, Model A is effective because acetylcholine is in the synapse. Model B is not effective because acetylcholine is not in the cytoplasm of the postsynaptic cell. So if you listed those 2 points, you would have gotten the 2 points for Part B. I'm moving real quick because we are just about to run out of time. So again, these will be linked, but I want to go over the immune response one real quick. So we're going to run a little over, but it's okay. It's not like I have anywhere to be. Okay, so let's immune response. Okay, and this one's going to be a little bit longer. So in the immune response question, it's asking, describe the effect of inhibiting step 3 on the formation of pores and the release of interleukin. So if you remember from the actual question, step 3 was when we had that gas dermin and there were some pores. So that gas dermin is what is opening the pores. If you get rid of step 3, the pores can't open, stuff can't get in and out. Part B said to make a claim about cleaving inactive caspase-1 results from the activation of caspase-1. A student claims that the pre-infection production of inactive precursors shortens the response time of a cell to a bacterial infection. Provide one reason. So the claim about cleaving inactive caspase-1, it removes the inhibitor domain of the protein. That inhibitor was that little black dot that was attached to it. Remember when it said inactive, it had a little black dot. So by cutting off that little black dot, that cut off the inhibitor, okay? Now, once you do that, then the cell isn't going to have to wait anymore to do transcription. It can just like go, and turn on the other proteins. Part C, a student claims that NF-κB protein is located in the cytoplasm until the protein is needed for transcription. Justify the student's claim with evidence. Identify 2 fractions where N-terminal gas dermin would be found in cells infected with pathogenic bacteria. So we're going over. So the justification, NF-κB and glycolytic enzymes are found together in the cytoplasm. So if you look back in a moment, let's assume you're watching this later when we can see both. That's the one thing about Crowdcast. I can't show you both of the screens. You can see both of those are in the cytoplasm. And then you would see that in fraction 3 and fraction 4, because again, when you go back to the actual question, you would see that is where those proteins are present. Part D, describe the most likely effect of gas dermin pore formation on water balance in the cell of a hypotonic environment. So hypotonic, what does hypotonic mean? What does hypotonic mean? Low what? Less solute. So in the environment, that means less solute in the environment, okay? So if you've got less solute, that means the water's going to enter the cell, and it's going to go floosh, and it's going to flood it all in. Part E, Explain how gastrointestinal pore formation and interleukin release contribute to the organism's defense against a bacterial pathogen. Cell lysis destroys the infected cells, or cell lysis prevents the bacteria from replicating. And then interleukin signaling will stimulate immune cells and components of the immune system to destroy the infected cells or bacteria. I know that was super, super quick and not super in-depth. That's one of my complaints about Crowdcast. I can't show you both of those at the same time, so I know that didn't look the best. But like I said, these are posted in the slides, and to make sure y'all can go ahead and see them, I'm going to go ahead and give you the slides. Woo. So y'all can go ahead and access that. We are a little bit over time, and I've got to hop off, but I hope that was super helpful and gave you the background information you needed in order to answer those questions. That was my goal through this. So I'm about to hop off, but it was super great seeing you guys. I hope this was helpful. Follow us on Fiveable. I'll be here next week talking about, what am I talking about next week? Next week, I'm talking about, enzyme catalysis. So we'll be talking about the enzyme labs. So tune in next week for that. Same time, same place. We'll talk about more FRQs. You'll get more resources from me. Otherwise, it's great to see y'all, and I hope y'all have a great rest of your week. Bye-bye.